Unlocking the secrets of rare disorders, the role of IKBKG mutations. Imagine a world where a tiny genetic glitch can lead to a cascade of severe health issues, yet presents so differently from one person to another that even doctors struggle to connect the dots. That's the reality for many families grappling with IKBKG mutations, and it's a puzzle that researchers are determined to solve. Every human body is a complex machine, with countless parts working together to keep us healthy. At the heart of our immune system's defense mechanism lies the NF-kappa B signaling pathway, a vital network that helps us fight off infections, heal wounds, and control inflammation. This pathway's key player is the NEMO protein, encoded by the IKBKG gene. But what happens when this gene malfunctions? The result is a spectrum of rare and often debilitating disorders, each with its own unique set of challenges. The journey from a genetic mutation to a diagnosed disorder is rarely straightforward. For the IKBKG gene, this journey is particularly convoluted. On January 12, 2025, a groundbreaking study published in the esteemed journal Genes Diseases shed new light on the clinical landscape of IKBKG mutations. This comprehensive review, led by researchers from Chongqing Medical University, analyzed an impressive 144 studies and 564 cases, creating one of the most detailed clinical maps of IKBKG mutations to date. The study's findings are a beacon of hope for clinicians, offering a clearer diagnostic framework for recognizing these complex conditions rooted in NF-kappa B dysfunction. By linking specific genetic mutations to a variety of rare disorders, the team provided invaluable insights that can help improve early diagnosis and personalized care. The review classified how different mutations correspond to conditions such as incontinentia pigmenti, IP, anhidrotic ectodermal dysplasia with immunodeficiency, EDA-ID, immunodeficiency, ID, and the newly identified NDAS. One of the most striking revelations from the study was the wide range of disease presentations driven by IKBKG mutations. While most cases were diagnosed as IP, 78%, followed by EDA-ID, 15.8%, ID, 5%, and NDAS, 1.2%, the symptoms varied greatly. Female patients predominantly had IP, while males were more likely to develop EDA-ID, ID, or NDAS. Common symptoms ranged from skin pigmentation abnormalities and dental defects to recurrent, severe infections and systemic inflammation. The study's findings also highlighted the importance of the zinc finger ZF domain of IKBKG, which is particularly prone to disease-causing mutations. Variants such as E390RFSX5, H413R, and X420W were associated with multi-organ involvement and high phenotype scores, indicating severe outcomes. Interestingly, some mutations, like E390RFSX5, appeared across multiple disease categories, complicating diagnosis and treatment decisions. Beyond symptom mapping, the study identified patterns in infection susceptibility. Mycobacteria and streptococcus were frequent culprits, and immunoglobulin abnormalities, especially IgG and IgM deficiencies, were common among EDA, ID, and ID patients. These detailed genotype-phenotype correlations offer a roadmap for clinicians navigating ambiguous or overlapping presentations, helping them to recognize cases earlier, improve outcomes, and reduce diagnostic delays. Especially in children, the implications of this research are far-reaching. By linking specific IKBKG mutations to clinical outcomes, healthcare providers can better anticipate disease progression and tailor treatments. Whether through immunoglobulin therapy, stem cell transplantation, or emerging gene editing technologies, The study also underscores the urgency of recognizing underdiagnosed cases, particularly in infants and young children with unusual infections or skin and dental anomalies. As gene therapy research accelerates, this comprehensive mutation map may serve as a reference point for designing mutation-specific interventions, bringing hope to patients and families affected by these complex syndromes. But here's where it gets controversial. Some experts argue that our current diagnostic tools are not sensitive enough to capture the full spectrum of IKBKG-related disorders. What do you think? Are we underestimating the true prevalence of these conditions? Share your thoughts in the comments below.